And usually the lunch break runs till 2 p.m. But today we're going to start at 1:30 with Stuart Russell's talk, he will be virtual. And because of time constraints, time zone constraints, we have to start at 1:30. So make sure you come back here in the room by 1:30 p.m. All right?
Thank you. So I'll continue. And this is the slide I missed in the earlier talk. So this is three image data sets. And the PCS very expensive. You do bootstrap. You can see this is in minutes. And this is using dropout. So you feed one model, you create different, instead of bootstrap predictions, you create different prediction by dropping out of the weights. And this is adding noise. This is feeding, embedding, more sophisticated. For each embedding layer, you feed a logistic regression type of thing. So you can see that for these two, we're basically quite comparable in terms of compute time, and we improve the sizes, right? So this is the best one, use 6.5, original almost cut into by 40%, and this is still a big, like this is a good like 20% or more cut, and similarly. So this is just showing for deep learning, we cannot do bootstrap. So we're finding different predictions by altering one deep learning, either through adding noise or by dropout. So I think most of people were here for the first part, right? So just three principal data science. PCS means predictability, computability, stability. And we intend to synthesize and unify. It's an evolving framework, and we want a uniform platform language and also ideas and principles. I like basic principles because that's how I remember things. And this is an attempt to really synthesize the experience as an applied statistician into forms that you can keep going back. And we want to look at system point of view. You have a whole data science lifecycle. It's not just one modeling step. It's every step. For this to work, you need to look at them as interconnected components. And you want to shake reasonable every step. You don't want the system to break. And what's reasonable depends on context. Okay, so for the second half, I'm going to talk about the development of iterative random forest. This was one of the two scientific collaborations that motivated the framework. Everybody heard of random forest, but for interpreting random forest to get gene-gene interactions, for example, nonlinear interactions, it's not stable. So we added stability. And then we use Drosophila data. And then we went to humans for pretty complicated human disease called HCM. And then we have some mathematical analysis for theoretical version of the methodology. And now I'll leave with my open question kind of thoughts. So it's really PCS guided machine learning algorithm. Actually, this algorithm development motivated PCS. So I'm just kind of going backwards because I already told you PCS. So in biology, instead of pattern recognition with a cat, we don't know what the cat looks like. And we find something meaningful, we don't know what it looks like. So there's a lot of search going on. Then I know we're looking for a cat, I just look for it. So it's really a combinatorial search problem. Traditionally, starting from Fisher, we know genes and genes work together. It's not just the independent work. That's how we are as we are. And people use traditional polynomial interaction. They go say, what are the important genes marginally? And then they look at polynomial interaction and keep adding it. But this, for many reasons, combinatorially, this is too expensive. And often in biology, now we understand better than 100 years ago when Fisher proposed this model. Some genes, many of them, independently not important. It's only in interaction with other genes they become important. So both scientifically, this is not the right approach. And at least for the problem I work on, and also computationally, it's too expensive. So this work was PNS 2018, and we want to really look at more than two-way interactions. And Samantha is now tenure professor. Cornell, Carl, you have seen. Ben is a colleague at IBL and in my department. So we have this particular problem we work on is something called enhancer. Enhancer is something to help a gene regulation. And the unit is like a segment of DNA. And we have data with properties of this segment and try to predict the status, enhancer or not, to simplify in the supervised learning form. And it's known that random forest predicts really well about the status. But then this is only association. If you really want to know which these properties of this segment really determine this status, you want something more trustworthy or mechanistic interpretation. And that's where we embark on this project. It's knowing random forest is working for prediction, kind of P is satisfied, but we want to see whether stability can help us to get to something mechanistic and interpretable and science. And we know that I work with Drosophila biologists. There are fourth order interactions. So this is the Drosophila fruit fly embryo, like after 2 or 3 hours after the inception. And then this is the lines of the size. This is well famous genes called bicoid, humpback, giant, and cripple. The position where they express themselves, you can see you need 4 of them to decide on the position. This is basically embryo state. That's why our spine, corresponding to when our spine developed. And Drosophila is really star biology, simple to breed, and has won the work on Drosophila has won like 5 or 6 Nobel Prizes. So it's a very, very effective modern organism. And the question is, why we like random forest? Why we didn't do deep learning? At the time, there was some deep learning already. There's this something called the French flag model, saying that for biology, one gene is not just one entity. It produces many, many little armies of things. And you need abundance. They randomly move around. Without abundance, nothing happens. So you need to have abundance and often reach certain threshold, interaction happens. So this thresholding behavior is mathematically mapped into these decision trees. You want these genes to reach certain level, basically the DNA occupancy, before you see things happen. So that's why we argued this is the right model. Post hoc explain why random forest works so well, because random forest is based on decision trees. And just a review of random forest, right? You do bootstrap sample, you feed card decision trees, you run the trees to purity, and then each time you do a split, you uniformly sample from all the features with a particular parameter called M trie, usually the same order of how many features, and then you average. So we don't really do the average step, we look at all the trees. And before us, people already thought about this is early work, like quite a few years ahead. They said, well, if you have all these trees, predict the enhancer status. If 2 genes fell or 2 predictions fell on the same path, and you make a jump, oh, they biologically interact because they seem to want to work together. So people did that, but things were not stable. You do that, but nothing seemed to stable. So that's why we came in. Actually, Ben Brown brought me to the problem. He was working with genomic more than I did. So what we did with Basu was a postdoc with Ben and me. We add stability to random forest, right? So you can see how predict satisfied, and we assess for interpretation is not stable enough. We're adding stability. How we add stability, this took a while, is random forest comes with it, something called MDI, mean decrease in purity. It's basically saying when you do the split, how much improvement you have on some loss function. And when you have big improvement, you think that's important because it really helps you. So with that, so first step, we do random forest, just uniform sampling. But for the second step, that's why I call it iterative random forest. We sample with weights based on the importance. That's a self-dimensional. Nobody's completely deleted, but we're concentrating on the important ones, but also allow the moderately important one to show itself. Hard thresholding, then you'll be deleting things. And then we took advantage of something called random intersection trees. Actually, that step can be replaced by deterministic algorithm, so let's not worry too much about it. And then we added outer loop of bootstrap. Random forest already have bootstrap. This is an outer loop bootstrap to collect some index about stability. And then we have similar computation memory calls as random forest. So in more detail, first, for each split, you have uniform sampling. This is already weighted something, it's already in this work. So we're putting different ideas together. And then for the second step, we have this MDR importance, we just weigh them. And then everything else is the same as random forest. And then we've added the after loop of bootstrap. So we mapped the, so in the end, for every step, we basically have all the trees. And the unit is a path. So a path on a tree has what other features is on the path. So you can code it as a 0, 1. So each path become a 0, 1 vector. And that become a similar problem as the bucket problem in computer science, right? You have two groups of people, blue people, red people buy something, you want to see what the common bought items, this becomes similar. So that's how the algorithm would tap into that literature to find the commonly bought items. Bought items for us is the shared features. Okay, so that's how we need a random intersection trees. So we end up with aggregating with stability score. So that's a search. So it's after loop bootstrap. See this gene 1, gene 2, how many times appeared in this aggregation of different features. Yes.
So are you trimming features when you do that?
What?
Are you trimming features? Are you ending up like with smaller sets of important features?
Yeah, we use stability to rank them. Yeah, we do.
Could you just explain the last line a little bit more detail? So what is the P, C, and S? S I understand, but I think the P and C was not completely
P means
clear.
a prediction check.
Okay, so what is RF there?
What?
Oh, the random forest.
Yeah, yeah. This is iterative random forest.
I see. So the prediction is using random forest, using the standard approaches to evaluate the accuracy, prediction accuracy. And the RIT is?
This is the random intersection tree. It's a computation because you have to compare which ones share the same like genes.
I see.
So you need a computation.
So that's the computation that you do.
Yeah.
Okay.
And then stability, it's a stability score.
Got it. Thank you.
Yeah. So go back to the enhancer prediction problem, right? What we have, we did 3 iterations. You can see our sequence basically in this 6. So we didn't lose accuracy. This is on test set. And here were some investigations. So here we have the stability score, just cut it at 0.5. And these are names of different genes or transcription factors. The red ones are pairwise interactions. And the blue ones are 3-way interactions. And we found 20 pairwise interactions. And we submitted paper actually to PNS, and the referee said, why shall we trust your interactions? Good question. I said, well, because Sue Seneca, who's a Drosophila scientist, says so, right? And then we went to Sue and said, why did you like them? And she said, here are the papers. People already done experiments before this high throughput age. People did old-fashioned ablation, use laser or something, so there were no interactions. And that prompt me now when I interact with experts, I don't give them only the ones we find, I give them 3 lists to avoid confirmation bias. The top ones, the middle ones, and random selected ones. If I have good collaborators, they shall be able, either we got it wrong, or they had faulty knowledge. So far, all my collaborators haven't failed. They always agreed with our top choices, right? Either we all bias together, but shouldn't be. So I give them 3 lists. With Sue, I did that. I said, oh, she liked it. I was like, oh, that's good. But we didn't record it. So that's why it's 80% accurate, because people already done before this work biological experiments. So now we have a more refined version. Before we only have whether 2 genes got the split. But we actually have information. Is the gene got split by larger than something or smaller than something? So we added sign. We call sign RF. Because biologically makes sense. What's abundance is like overexpression or suppressed expression, have different roles. So we have a more refined version. That's what we use, called the sign, we call SURF. It's similar, right? Just map into 0, 1, you have 0, -1, +1. So it's similar. That's the theoretical work we analyze. And then we worked for 5 years. Carl was gone already. We made this work for Drosophila into a browser for UC Santa Cruz genome browser. So also all other scientists could take advantage of result. And for this work, we had more predictors than the original one. And so it's much more expanded. And Sue Seneca, the Drosophila scientist, joined us, and another Drosophila scientist. So this took 5 years of work, and the paper never published. It's on ICA. But I think this is what I think we're most proud of, is because you try to bring it to the users. But it's not published. But that's the version we use, actually, in the theoretical analysis and also young work. Then the questions, Drosophila genetics are pretty simple. They are bred, and they're very pure. They're fed on the same food. It's a good test. Does it work for human genetics? Right, we're now fed on the same food, and we have all kinds of environmental factors, and we're not that pure in terms of genes. So that's the question. So similar, we want high yield experiments, and we have a new version called low surf. Low means low signal to noise ratio. This is just very, very, very noisy data. Again, this paper on the project finally got published, accepted by Nature Cardiovascular Research. Again, started 5 years ago or 7 years ago. So there was a call, Changzhou Global Biohub. And we organized a team with 3 UCSF, Berkeley, and Stanford. And I mainly worked mostly with these 2 guys. First, he's now with a startup. They both cardiologists. He's actually department chair of medicine now. Ewan Ashley, James Priest, and other people will help. And then, so the first human traits we look at, I won't talk about, is actually red hair. Red hair is a lot purer than heart disease. That was the first case we did, was mainly with James. And Merle, who is now back professor in Regensburg University in Germany, and Carl led that project. And what I'll present next is really the heart disease. So it worked for red hair. Red hair is pretty pure, it's genetics, and I don't think the environment might impact you as much about your hair as the heart condition. And then these 2 really bright young people have been the lead junior authors. Chen Ru is a postdoc with Ewan Ashley. She had mechanical engineering degree, PhD from MIT, and Tiffany Emanuel was my former student. I'm Notre Dame assistant professor. It's a huge team. So we wrote a proposal about another heart disease, and we couldn't find any signal in UK Biobank data. So we switched. We switched to this HCM. HCM is a specialty of Ewan Ashley, who is the cardiologist. And it's a heart condition that occurs in 1 in 500 people in the US. And the heart ventricle wall becomes thickened. And then therefore, it's hard for the heart to pump, and you have also smaller cavity. So both are not good for your heart. You can faint, you can have heart attack, you can die. And people have shown prior work that the cell size might be the reason the wall become bigger. So that gives a good phenotype. For the Drosophila problem, I had a complete failure, and Sue's lab for another project on Drosophila did 40 experiments. And we find a very important gene we couldn't publish just because we didn't predefine what's the phenotype. You alter the system, but you need to know what you're going to monitor. And that was not well defined. So I learned very expensively that you want to have something you can do the experiment and know what you're going to monitor. So we have that. It's the cell size. And Chen Ru already built a whole pipeline of sorting cells, all of that. So everything was ready. And then people believe that there's an important genetic component to it. Okay, so here we're really going for causality. We want to develop eventually drugs. And causality for me is not yes or no. It's accumulation of evidence. There's a philosopher called Nancy Cartwright from UCSD who said that causality is when you have enough evidence to act. That's my definition. Act for me is to ask you and people to do experiments. Because you're getting other people to do work and it's expensive. why are you going to say that there's enough evidence to act? And FDA act, they look at Vioxx painkiller, they look at evidence, they decide whether the drug will be released or not, right? That's their act. And even in randomized clinical trials, you have to make an assumption called suitable. Basically, you have one version of the drug and there's no interference. So there's no way of getting causality without assumption in the statistical world. In the physics world, you have Hooke's law, you put a load at the spring and the length get bigger most of the time, and you can get unlucky and then it breaks and then the law doesn't hold either. So it's kind of mechanistic, but it's not 100%. So what we try to do with PCS is predictability follows from causality. Stability also follows from causality. So if there's really causal, you hope different machine learning methods will show that they're important. And then we come up with interval hypothesis generation recommendation for experiments. So I don't claim causality from observational data. I become a recommendation system toward cost-effective design. If you compare with the traditional approach in causal inference based on observational data, as I said, lung cancer caused by smoking was a series of observational studies that there was a human decision is causal. There was enough evidence to act. That's why all the labels on cigarettes and all of that. So they don't usually do a lot of model checking. They assume propensity score basically after you condition a certain covariates, nature will toss a bias coin to assign to the smoking group or non-smoking group. Or for us, HCM group or not HCM group. So we use prediction screening, try to do some model checking. And then they basically use domain knowledge to basically say, well, the propensity score is really probabilistic because you factored out all the confounding factors and you have a toss. We do stability analysis. It's very algorithmic. And we don't claim causality. We claim, hopefully, trustworthy hypothesis generation for experiments. And the causal people will usually just claim causality. So this is a comparison. So the goal is we want cost-effective design. And epistatic genetic interaction is actually the frontier of functional genomics. GWAS, you probably have heard, right, has been going on. And then the single important genes, they found a lot of good ones. So finding interactions which is a lot more subtle. And that's the frontier. And we think, this is a belief, it's not a proof or theorem. The reason is because we look at expanded account of uncertainty under PCS. So when we find something, that's more trustworthy. That's a high level belief. And the few case study we did really support that. So what's the data? The data is UK Biobank data. And something called SNPs or SNPs is a single, what happens that there's a common, this is a location on your DNA sequence, right? Single nuclear variant. And what recorded here, suppose this is me, it says that you have two strands of DNA that differ from the majority in both. You get the two. If you differ only by one, you get a one. So it's really saying that how different you are from the majority. And that's likely explained because most majority are healthy. That's mostly maybe explain why you might be sick. So that's the SNP data. So the data is actually not all observed, a lot of imputation too. So it's not independent. So we started with the label from UK Biobank. This is we only use unrelated individuals. This is usually older people, 50s and 60s, volunteer to have their data shared. And we couldn't beat the random toss of 50%. Hindsight, this is a couple months, right during the pandemic. It's because a lot of people have HCM or not diagnosed. So a lot of false negatives. And then we went back to a smaller set of data with people with MRI, heart MRI. So again, phenotype. Then we use somebody else's deep learning algorithm to find the volume of the heart wall of the left ventricle. And then we normalize sensibly by heart and weight because big people have big volume. And we have a sensible way to normalize it. And so this pipeline, I think, can be used for other UK biobank cancer phenotype and other disease phenotype for low signals. And then we have a regression problem because this is a continuous response. And then the question is, how do I know I'm not getting noise? I have no idea about the noise level. Thinking about, we need to have p-screening, we need some reality check. We binarize this continuous variable into the top 20%, so also clean the signal. The one is big heart and small heart, because it's easier to differentiate. And we did 25%, 15% to show that the results are consistent. And we also need dimensionality, not 15 million. So we use GWAS to screen the features. And we beat the random toss by 5%, 55%, consistently across different methods. I've never done a supervised learning problem with such a low prediction rate, but if you think about how complicated the problem is, you probably shouldn't expect a very high, because so many environment factors, right? We're looking at the DNA level, there's protein, there's transcription, there's so many steps before it becomes the phenotype, and environmental impact, the diet, all of that is not taken into account in the DNA. So you can also say that probably that's why you should expect very low accuracies. That's why you need to do experiments. So here's a pipeline. We randomly split the people with heart MRIs in the UK Biobank into the following form. And then we did binarization, think like scientists. Scientists usually go for clear signals. So that's what we did. We made the heart, big heart people, small heart people relative to height and weight. So simplify a problem. And the impetus came from, because I was working on another project with binary like crazy. So just it's in my mind and seemed to make sense here. And then it's a very weak signal, right? We try different methods and consistently we get around 55. The reason we went for random forest because of Drosophila success, the match of decision trees with the French. So domain knowledge came in. We use interpretation from random forest, and all the other methods we don't like the polynomial interaction. So a lot of prior information went into, we use SIR, random forest, stabilized random forest to interpret. And there's something else. When we did the screening, GWAS have two popular softwares. And we use both. And then we did a union, because we don't know which one to trust. But they only overlap 50%. So we end up with 1,500 genes instead of using one would be 1,000 genes. So we took the top 1,000 and did a union. So we have stability relative to the two software packages. Yeah.
So actually, I have two questions. One on this last point. So why the union in particular?
Because we don't want the software, the first step, to bias our results. We want to be inclusive in the beginning.
So a bigger set is better than taking looking at the overlap between those?
Because they're all marginal kind of methods.
Oh,
we
I
are
see.
going after interaction. Right?
Oh, I see. Okay.
So any one of them, they have different linear model type random effect assumptions.
Mm-hmm.
And we don't want one of them eliminate possible candidates for us.
Okay. And on the previous slide, I didn't understand the last sentence you had back there. The dimensionality reduction is necessary. Could you explain that?
It's because we first cannot recommend like 50 million of them, right? Only a few.
Sure.
But it's also the random forest doesn't run very. Either random forest or random forest doesn't run very well if you go beyond a few thousand.
Oh, I see. Okay.
Yeah.
Right.
Yeah.
Thank
That's
you.
what I mean. Yeah. It's a little cryptic. I agree. And then we recommend interaction by like stably important for binaries for different O3 binary thresholds. We did 15, 20%, and 25%. And then we did the wild-type gene silence experiments. So the takeaway is that for this very complex problem, data science is not enough to really settle the score. It's a multi-step evidence collection process to boost the signals, including wild-type experiments for trustworthy causal discoveries. So we have to reformulate the problem, right? So right there, use the label didn't work. And we developed a new method for surf. What happens is that if you look at each random forest at the SNP level, nothing is stable. So what we did is that we mapped each SNP to a gene. And when we run the data at the SNP level, when we collect the interpretation, we collect it at the gene level. So SNP 1, SNP 2 seem to be on the same path. We map into gene 1 and gene 2 and collected gene 1, gene 2 as the final result. And for the red hair, there was a biologically inspired software that can map us into genes, but we don't have it here because the cardiologist said that pipeline was built on dead tissue. And he believed for heart and brain, the electric signals is so important. Dead tissue, this featureization, he doesn't trust. So we were forced to work with SNP data directly, have to come up with more mathematical ways to aggregate, we map into genes. Otherwise, still even with random forest, it's not stable because the signal is so low. And then we did tons of annotated database search. It's like their databases show, oh, this gene might have this functionality, but nothing about interaction, so about single genes. I did some, we worked with a guy called Wes, who's a young fellow cardiologist, and then we did gene silence experiments. And then we really engaged with them, help them to improve the protocol too, because there's some huge, I'll explain. And another whole year, find other data analysis, actual data source, and then discussion with referees. They want us to repeat everything in the conventional way. And the first response to the first review was 38 pages. So this is huge amount of work. Really a shout out for my two young co-authors, Chen Ru and Tiffany. They never complained, said, I want a quick paper. Just do it, do it, do it. Really hats off to them. So Chen Ru is a mechanical engineer. So she built this machine. So you force all the cells, right, in our body, our muscle cells, heart is like that. But this is like a stem cell, it's all round. So the size becomes the diameter. That's the best we can do. The next phase is a mouse model or something, but we're not there. And we did gene silence experiment because it's cheaper. Instead of CRISPR-Cas9, you knock it off. So this is silencing it. And then she built this machine, you feed all the cells, and then we sort it and automatically find the sizes. And this is the undergraduate who prepared the sample. And then when we work with them for the data result, we actually discover some huge cell because it's automatic segmentation. The huge cells got counted like 40 times, got move very slowly. And they were very happy. Actually, in the beginning, they were not too happy that we want to look over their shoulder because they have used it for many years. And then they were very happy because we really vetted every step. And then they changed the force, so break it. And we also do robust statistics, delete the outliers. Because the big cells, we're looking at cell size, they're actually clumps of cells. They just didn't separate and multi-counted. So all of that's like old-fashioned detective work. So we did 4 sets of experiments. Again, 80% that's by chance as the last set of for Drosophila. We found 2 interactions. So this is a titin gene, it's well-known a heart muscle gene, and this gene is next to it. And this is a growth factor gene. So all related to the size. People thought they just always have the same functionality, and what we learned is actually they don't. They sometimes work together, sometimes they don't work together. So that's a new discovery. And after many like rounds of 3, and the paper accepted by Nature Cardiovascular Research, and another shout out to our main co-authors. And the paper is now 88 pages or something. But the hope is that somebody will develop a drug for the heart disease. And James Priest, my collaborator on the red hair, he's now leading a research group in a startup. So I've been talking to him to see whether I can convince him to invest, do a mouse model and clinical trials. But I convince him. I also try to say that you need careful work to really lead to something like a drug. Seems very long, but we did all conventional analysis and use annotated database show that they don't find as many relevant genes as we do. So that's where the referee convinced. Okay, so I think this pipeline is useful. We have all the codes there. So now I have very little time for the statistical mathematical, but let me just give you. So first, what's the relevant stylized models? And all the random forest work is assuming smoothness. We're not going after smoothness. We're looking for Boolean interactions. So then the raw models. We come up with a local spiky sparse model, which already used in the first E3 random forest paper as a simulation model. So the model actually was, and we did a theoretical version RF and proved some asymptotic model selection consistency for the interactions. And we did some simulation study. So this is the model. It's a regression model. We assume that you have continuous parameters and you have these Boolean interactions appear in decision trees, and you have different thresholds. So the gammas and the betas are continuous parameter, but we want the sets. It's not, if the axes are with overlapping interactions. So the first analysis we did is that all these interactions are not overlapping, which is definitely not true biologically because different pathways share genes, which is the first cut. And if you allow overlapping, there's identifiability issue. You have to redefine things, right? So you just put another block here, and one minus that, they're the same model, but they give you different interpretation. So we have to simplify things now, right? Reweighting is hard to analyze. So we decide that instead of allowing arbitrary threshold, we're just going to cut when the drop of MDI in pure hours is larger than a threshold. We introduce stability that way instead of through the reweighting, all of that. So it's really a theoretical version. And the interactions actually can be replaced by deterministic algorithm. We just remove that. And we also worked into subsampling and bootstrap into a cleaner version randomly. So there's a lot of simplification for the theoretical result. So three randomness. Data randomness, we condition data, we remove that. Tree randomness, growing individual tree, so we deal with that. And we have a simplified random forest construction. And there's also path randomness. So we also have a simplified. So basically simplify, idealized these three levels of randomness. So we can do analysis. Yes?
What path randomness, what is the path that is referring to?
If you think about it, right, in the end, I have all these trees and a lot of paths, right? So empirically, you just collect this path. So instead, we're basically just saying that I start from the node and I have certain probability to create the edge. So that's, we're saying this is kind of equivalent to the way we analyze it. So this become a random walk type of thing. Yeah, that's what I mean. Good question. Yeah. And then we define stable path. This is really saying that giving a tree and a path, tree and path both have randomness in that. The data doesn't because we condition data. This gives the index corresponding signs of first appearing stable features with empirical index larger than threshold. I know it's a mouthful. So you look at the path and you collect this index with sign, depends on which way you split. And the first time this feature appears, so make it unique. So it's really collecting which features get touched upon and seem to be important. And for any, so stability with through a form of prevalence. For any sign feature set, the features that this got split down, this is less than something, this got split down, this is larger something, that's what the second sign means because we can split both ways. And for any given such a sign feature, we look at the probability, the depth weighted probability. Remember, a path is through this symmetric split, and this gives me a probability, weighted probability. And then you can prove that for any such a sign set, there's an upper bound. That's the size with Gaussian additive noise. We need non-overlapping basic Boolean sets, independent features. This is all very basic. And then we need the number of features selecting order of P. And then you can prove a lower bound, showing that if the set is not the exact union of the ones appear in my assumed model, then there's a gap. So that's the upper bound, and it will be significantly real less than the upper bound. So when you exact getting the union in my true model, you basically asymptotically reach the bound. Otherwise, you have a gap. And that's how we devised what we call LS fine. This is the theoretical version of RF, right? You basically just say, do you reach the bound with some threshold to turn asymptotic into a finite sample algorithm? It's really just looking at the bound. Very, very delicate. So, and we can show that without ever estimating these continuous parameters, we can get at Boolean interactions. And that's something I kind of already believe by going to BioHub talks for like 2 years, 3 years. The biology figure out a lot of this high level logic type of relation without doing computation models, logic thinking. And I find a paper by a guy called John Paulette, a chemist in 1964 called Strong Inference. He was basically saying what I believed too, is that the continuous parameters are flimsy, but the logic reasons are more stable. So this is really, in hindsight, we went for the most stable bits of information first, without ever worry about these more flimsy parameters. And one simple example to show this, if you have uniform 0 to theta, you do max likelihood, it's the max of the order statistics. You can estimate the theta 1/n rate. In Gaussian models, 1 square root of n. That's kind of indication that these discrete structures are easier to estimate than the continuous parameters. So we kind of show that. It's kind of model selecting consistency you can call certification, and the RF doesn't estimate parameters either. So what we learned is that higher order interactions under this model are very hard because you only appear in the region minus L, that's the order. So you have very, very little probability to observe that. And this is what people do anyways. We show that you need the order P. And the hard thresholding works well for theory, but it's too strict. And you look at RF, we don't have a threshold. The weak signals actually can appear when you don't force a strong threshold. And that kind of pans out in some simulations, right? This shows that you have order 2, order 2 interactions. In truth, you only have 1. And you can see that when the higher order is getting harder, this is the correct model. When model misspecified, things get harder. So basically that's what, and then you compare with the blue itself, sorry, the color swapped. And then the orange is the theoretical version. You can see that when you have different signal to noise ratio and different correlations, here, the theoretical version just doesn't work at all. But the RF still find things. So I already said that it's a pretty ambitious program and it's evolving and we want to build trust. And I was at Geneva Farm 2 years ago and they asked, what's the secret sauce? It's really data science hygiene and a very common sense idea that you get multiple genes from good doctors and integrate. That's basically what's going on. Right? You have good experts, which is good algorithm, prediction checked, and then you just aggregate some way. And large language model development, right? This is in my textbook. You have data. All of this is question mark. What's the data? We don't know. What ChatGPT use? What algorithm? We don't know. What's future data? There are many possible ways. What's reality? What's decision? So it's really you need new thinking. And we have a paper with my colleague here, Ala Mahaman, to really look at from the PCS lenses. And we have some recommendations that you can look at. It's a 4-pager. It's really saying that what's reality check, right? I think we need to get them opened up ideally, at least for medical. So user can have user cases for them to test. Right now, it's all closed form. I mean, even during the war, some scientists got the top secret kind of clearance. I don't see why this big IT can give some people some clearance for them to have some oversight. I don't see why it cannot be done, but there needs to be effort. And computing is a bottleneck, but NSF have two platforms I've been using for my bootcamp on research. And here again, it's very easy to use stability. People already do that. They call consistency. You give the same prompt multiple times and look for consistency. Reality check is harder. So stability is not hard as reality check. You need external something or RAG. And I think we need some FDA-like thinking and documentation, right? For a drug approval, you have to write the SAP, statistical analysis protocol. We need something, but FDA is already proving a lot of AI algorithms. And I think the guidelines pretty sketchy, and we need documentation, and also not just the downstream and upstream. So we have some software people can take advantage of. And we have this high quality simulation kind of position paper we call merits, to one maybe related to the formal analysis, one thing to be modular, efficient, realistic, and intuitive, transparent, and stable. So I think all of this could maybe be combined with what you guys think about. And documentation. We have a template, the book. And vdsbook.com. It's free. So we're still finding typos. So easy to correct that version, but not this version. How is different? We try to mirror data science lifecycle. It's really bring liberal arts thinking, qualitative thinking together with quantitative thinking, and PCS every chapter and comprehensive. It's really try to get the critical thinking going. We don't assume a true model. We come from practice and tell students what you face. You have multiple choices. How do you make sense and collect evidence? And we have 5 kinds of exercises, codes on GitHub and data set on the GitHub. And we got a very positive review from UL and UL Benjamini. UL Benjamini is the fame of FDR, false discovery rate. And with the inaugural book review from Harvard Data Science Review, disclosure, I became a co-editor. Chief editor wrote a very nice note summarizing the review, called it Pedagogy Excellent, Diverse Examples and Projects. It's a valuable source for data science in general, not just for data science students and for domain. Very little mathematics notation used because there are a lot of great mathematics books. It's very accessible. A colleague gave it to some friends of her high school kids, and they seem to be able to access it. So it's just common sense. Who should reject reality check and checking on arbitrary kind of reasonable human judgment calls? So where we're going now, we're deeply working with doctors, cardiologists, and also ER doctors. I believe medical AI, so area AI will have positive impact, and also we need safety. And we look forward to Friday. We work a lot on deep learning interpretability, mechanistic interpretation, and we continue with PCS and certainty quantification and use it for active learning and other ways of doing things. We want to go to Rome. We have a workshop in Rome in June 20th, and we're still accepting. If you're in the region, consider we're still accepting contributed talks. We did one with Stanford last year. It was one day, it was very successful, and we're doing one in Rome on June. It's a Friday. So if you're in the region, open problems. I think mathematical certification only possible at the modeling stage. I'll say that. So conjecture, statistical certification with positive probability for my particular model, I don't think you can get asymptotic consistency when you have dependent features and you have overlapping, but maybe with positive probability. I think that might be likely. And I'm very, very interested in a synthesis of different notions of stability. I'm really in love with stability as a general principle. And there are different notions of stability in the literature. Wouldn't that be nice to synthesize that and look at their different proof techniques, different conclusions, and make it a central consideration in theoretical analysis? And the empirical sets of PCF indicate that we should be plural. We should not just use one model. And it'll be interesting to see whether your certificate verification, when you combine from different angles, our conjecture, I'm convinced that you'll do better result in practice. And how do you formulate that theoretically to show that? We have a little bit such result to show that if you have different class of models with Ming-Xie, that we can show that under some conditions, the PCS, the new form, PCS form of conformal inference will give shorter intervals than the existing conformal. And going back to Sanjit's talk, what we can come out as a reasonable world models specification for the data cleaning step. Right now, it's very holistic, but there are softwares to do data cleaning already. Are they satisfy what we wanted to satisfy? Similarly, what are reasonable world models for the explorer data analysis? People look at data, right? And with my student from CIAT, they also were building some visualization to give people the option to try different opportunities, right? Hopefully, but what's the formal certification for visualization step, which is very, very important for explorer data analysis. And when you go to verify systems, how the PCS principles requirement mesh with what you guys come up with. As you hinted there already, I think formal analysis there has to be a combination of mathematical results and the reasonable world models and multiple data-inspired simulation models and results. I don't think mathematics can really solve. The question is how we can put into some boundary conditions for the simulation and what type of probe specification for this model to generate. And so together we can never show 100% guarantee, but we can really improve the safety parameter of any system. Here's a list of papers. Thank you. So watch out for my website. We're finishing this paper for the uncertainty quantification.
Right. Thank you, Ben. I think we have time for maybe one or two questions.
So I want to emphasize again that right, world models are needed, but they're very hard to get. Verifiable ones, but available now are evaluation models, right? Part of the machine learning cycle is that we have to evaluate our models. And plurality means that we're going to have model multiplicity, right? Different models are going to predict different things.
Yeah.
And so, for machine learning, right? Machine learning is not just decision, it's also evaluation. And so there are formal evaluation models.
Yeah. So I'm starting a project because benchmarking has played a huge role in machine learning and also deep learning right now. I think we need to move from overall accuracy to subgroup analysis minimum. So we're going to try to do something to show the ranking of models might not be so consistent if you look at subgroups. So that's about evaluation. And multiple metrics too, not just one metric.
Other questions? Right, if not, let's thank Ben once again. All right, so we'll break for lunch now and please be back here by 1:30.